Chapter 9 covers extranuclear inheritance, and what we're going to talk about here is any inheritance that does not involve the nuclear DNA. So you can get this usually through three different ways. The first is through organelles, and we'll talk about these, but there's DNA in mitochondria and chloroplasts. This is usually inherited from the female egg down to the progeny. Now, again, think about how it's set up. The female egg is usually quite large. It's going to contain most of the cytoplasm, the organelles that are needed, including these mitochondria and chloroplasts. In contrast, most sperm quite small, primarily only going to consist of DNA with maybe some proteins involved as well. We also can see infectious heredity. This results from a symbiotic or parasitic association with a microorganism that happens to be in the cytoplasm of host cells. And so they can exert their own effect on how different genes might be expressed. And then lastly, maternal effect. So nuclear gene products, things like mRNA or proteins, can be stored in the egg and then transmitted to the offspring. So looking at the organelles, it's a very complicated issue to analyze inheritance patterns that result from mutant alleles in the chloroplast and mitochondria. And there's really two reasons for this. First of all, the function of these organelles is dependent on gene products from the nucleus and from the organelle DNA. So it's not just dependent on its own DNA. Many of the functions also require nuclear products. And so finding the origin of mutations affecting these different organelle function can be quite difficult. Also, within a given cell, these organelles that are contributed exhibit great variation. We call this heteroplasmy. And so this makes detection of this mutation or analysis more complicated than simple Mendelian inheritance. Because imagine you have a cell and you have mitochondria and maybe 10% of the mitochondria are mutated. Is that enough to give you an effect? Is it not? How do you isolate those from the 90% that are fine, as well as from the nuclear genes? And so as you can see, it's been very complicated to study these. However, there have been a lot of studies looking at mitochondrial DNA in particular, sequencing it, trying to identify where these mutations are happening. So with these organelles, we have to talk about where did they come from? And where they came from is this endosymbiotic theory. And what it's thought is that way back, there was an aerobic prokaryote, there was a eukaryotic cell. It took up this aerobic prokaryote, and it was beneficial for the organism to keep it around. Imagine that this aerobic prokaryote was doing things like breaking down sugars for energy. Yeah, that would be very beneficial to keep around, and so you can see why it has been associated for a long time. Similarly, with the photosynthetic prokaryotes, if it was taken up, this is encoding genes that are going to do photosynthesis, and so making sugars for the plant. Again, very beneficial, and so it was likely to stick around for quite a while. Now, over time, many of the original genes have been lost, and only ones that are important for the various functions, either cellular respiration or photosynthesis, have stuck around. Also, both the mitochondria as well as the chloroplast have this double bilayer, lipid bilayer. And it supports the origin from this as originally coming from the outside plasma membrane as well as the prokaryotic plasma membrane. So chloroplast DNA ranges from about 100 to 225 kilobases in length, and the genes that are carried on this DNA primarily going to encode products involved in photosynthesis and translation. And we know this because mutations in these genes can inactivate photosynthesis. The mitochondrial DNA, or mtDNA, is smaller than the DNA in chloroplasts. We see that introns are absent. This is also something that we see in prokaryotic cells. They typically are not going to have these introns. And gene repetitions are rare, and so it's really been pared down to the most essential genes for this. Most of the protein encoding genes are located on a single strand, and replication in the mitochondria is dependent on genes encoded by the nuclear DNA. And so the mitochondria is basically not self-sufficient. In order to replicate the mitochondria, you need genes encoded by the nuclear DNA. And so they've really, like I said, pared down the most essential genes for this particular function. Because we have genes already involved in replication in the nuclear DNA, no need to replicate those within the mitochondria. Just makes it a little bit more efficient that way. Human mitochondrial DNA contains about 16,000 base pairs, and they code for about 13 of over 70 proteins required for aerobic cellular respiration. So as you can see, still many products that are required for this product coming from the nucleus. For cellular respiration coming from the nucleus. Mitochondrial DNA in particular is very susceptible to mutations. Some of the reasons why: it doesn't have histones, which are nuclear proteins, to help protect it from mutations. So within the nucleus, the DNA is going to be associated with histones, and this can help to protect it from various things such as reactive oxygen species. Mitochondria have very high concentrations of reactive oxygen species, or ROS. These are generated by cell respiration and basically involves free radicals. This is going to damage the organelle contents, things like proteins, lipids, mitochondrial DNA. Again, it's fairly helpful for it to be encased within the mitochondria so that these reactive oxygen species are less likely to go out and attack other products within the cell. If you remember back when we talked in Biology 111, we talked about cellular respiration process. There's some electron carriers involved in this process, and really the transport of electrons incredibly important for the cellular respiration process. And so these can be formed, can really just damage or attack different things within the mitochondria. Studying this mitochondrial DNA can really help provide insights into, first of all, the critical importance of this organelle just during normal development and normal function, as well as the relationship between mitochondrial function and some different disorders, neuromusculatory, the neurological disorders. And in fact, defective mitochondrial DNA is really implicated in the aging process. So almost all major human disease conditions really have the mitochondrial dysfunction involved in some way. And these can include type 2 diabetes, arterial sclerosis, neurodegenerative diseases such as Parkinson's, Alzheimer's, Huntington's diseases, different brain disorders like schizophrenia and bipolar disorders, as well as many different types of cancers. This aging tissue that we find contains mitochondria with increased levels of DNA damage. And so it's just thought that as we age, these reactive oxygen species are attacking the mitochondrial DNA as well as other products within the mitochondria, causing it to not function as well. Then lastly, we can talk about the maternal effect. And what happens here is you have phenotype for a trait under control of nuclear gene products present in the egg. And by nuclear gene products, primarily we're talking about proteins or untranslated RNAs that might be present in the cytoplasm of an egg. And what you can see here is this is a Drosophila embryo, and they have a particular mRNA. It's very concentrated in one region of the Drosophila embryo, and you can see there's just this gradient as development takes place. And so very early on, because it is such a high proportion of the embryo, it has a very large effect, and then as time goes on, as the cells divide, this effect becomes less and less because it effectively gets diluted each generation of the cell division. So this week is a fairly short week lecture-wise. What I want you to concentrate on is making sure you're all caught up on this week's quiz as well as all previous quizzes. Make sure that you're up to date on the homework, including the Mastering Genetics and any other handouts. Make sure you're keeping up with discussion forum questions and including interesting insights at least once a week. And then lastly, really start thinking about your student projects. You should really have a title, a general topic that you're going to focus on. Start to outline what you're going to do as well as pick a presentation style. Really, this should be done by about the beginning of spring break. And I would like to see what everyone has worked on so far. If you need help picking a topic, narrowing things down, working on the outline, please see me. I'm very willing, very happy to help with this wherever I can.